Good afternoon, everybody. My name is Zachary Barrera, and today we're going to be talking about the chemical disulfoton. So disulfoton is a selective systemic organophosphate insecticide and archaicide that is especially effective against attacking insects. Its molecular formula can be written as C8H19O2PS3, and in basic terms, this chemical presents several levels of toxicity and poses as an environmental hazard. It is a manufactured substance that is used as a pesticide to control a variety of harmful pests that attack both field and vegetable crops. It's primarily used in agriculture, but can be used in home gardens as well, with its use decreasing in recent years. It is a dark, yellow, oily liquid that contains an odor similar to that of sulfur, which is pretty strong. So moving on to our next section, we're going to talk about the toxicokinetics and toxicodynamics of disulfoton. So toxicokinetics describe how a toxicant enters the body and reaches a target tissue, while toxicodynamics describes what happens to that tissue once the toxicant reaches an effective dose. So data illustrates that disulfoton is readily absorbed by the gastrointestinal tract, and the urinary metabolites of disulfoton, bear with my pronunciation here, are diethylphosphate, diethylthiophosphate, diethyl-dithiophosphate, and diethylphosphorothiolate. Human urine isn't always indicative of the presence of disulfoton, but could also be several other organophosphate insecticides. Studies in lab animals support developmental toxicity as a sensitive endpoint following oral exposure to disulfoton. Following oral exposure to both parents, rat offspring showed significant inhibition of brain or red cell acetylchlorinoesterase activity. Sorry for my pronunciation, these are some long words. So, along the terms of acute toxicity, disulfoton is very highly toxic to all mammals by most routes of exposure. Whether absorbed through the skin, ingested, or inhaled, early symptoms in humans may include blurred vision, fatigue, headache, dizziness, sweating, tearing, and salivation. It inhibits choline esterase and affects nervous system function. Symptoms occurring at high doses include defecation, urination, fluid accumulation in the lungs, convulsions, or coma. If doses reach an extremely high amount, it can stop respiratory muscles or constrict the windpipe. Some chronic toxicity studied in rats show that they have survived daily doses of 0.5 milligrams per kilograms per day for 90 days, while other studies have shown that rats can acquire a tolerance for the chemical. In a 2-year rat study, males that were fed disulfoton, daily levels at below the LD50 had increased spleen, liver, kidney, and pituitary weights, while females with similar treatment had decreased weights in these organs. Also, at all dietary levels, male brains decreased in weight while female brain weights increased. At the highest doses, choline esterase activity was inhibited in both sexes in the brain, plasma, and red blood cells. So the image depicted in the background is actually a 3D model of disulfoton. And while it's kind of hard to tell, there's actually a total of 32 bonds, and they consist of 13 non-hydrogen bonds, 1 multiple bond, 9 rotatable bonds, 1 double bond, 1 sulfide, and 1 phosphate. Not gonna lie, trying to find exposure limits for this particular chemical were pretty tough, but I was able to find some data. The National Institute for Occupational Safety and Health recommended airborne exposure limit of 0.1 milligrams per meters cubed averaged over a 10-hour work shift, while the American Conference of Governmental Industrial Hygienists only recommended an airborne exposure limit of 0.1 milligrams per meters cubed averaged over an 8-hour work shift. So their time frame is about 2 hours shorter than that of the NIOSH, but that only accounts for the air levels and doesn't account for levels of disulfoton ingested through the skin or any other way other than airborne exposure levels. I was not able to find data on dermatology for this chemical. So I briefly hit on this topic a little bit earlier whenever I was discussing the background of disulfoton, but primarily the use of this chemical is to protect agriculture and home gardens. So in agriculture, it is used to protect small grains such as sorghum, corn, and other field crops, some vegetables, fruit and nut crops, and ornamental and potted plants against certain insects. And although it is used primarily in agriculture, small quantities are used on home and garden plants for mosquito control in swamps and other small insects in that regard of home gardens. And the use of this chemical actually has decreased in recent years, which I also spoke of previously. So disulfoton actually is a manufactured substance that doesn't occur naturally, but I can talk about some places it can be found and how it interacts with the environment. So it can be found in soil to some extent as it has a low to moderate persistence in soils. It is not strongly bound to soil, and some metabolites are more mobile than the parent disulfoton in sandy loam, clay loam, and silty clay loam soils. Mobility decreases as organic matter content of the soil increases, and in addition, these metabolites can persist longer than disulfoton. In a study done on sandy loam soils, disulfoton had a half-life of 1 week and a 90% loss in 5 weeks. One metabolite had a half-life of 8 to 10 weeks, and another was fairly stable for 42 weeks. Disulfoton has been found in groundwater in Virginia and Wisconsin at levels up to 1 milligram per liter. So its breakdown in water, like other organophosphorus insecticides, disulfoton will break down in water under alkaline conditions and is most stable at normal surface water acidities. The degradation of the compound is also temperature dependent. So its breakdown in vegetation, when applied to soil, disulfoton is actively taken up by plant roots and is translocated to all parts of the plant. Such systemic distribution is especially effective against sucking insects, while predators and pollinating insects are not destroyed. Control may persist for 6 to 8 weeks, and breakdown in plants follows the same chemical pathway as in the soil. Well, that'll wrap up my presentation over disulfoton, the chemical. I hope you all learned something today, and above here is my references that I used for this project, and Tom, I hope you have a great winter break. Thanks for a good semester in this class and in Sources and Fate, and I'll see you soon.